have found like 3 bands for NAP, right?
Yeah. Yeah, I didn't quite get 3, so not exactly sure what happened.
But you're not getting that tail effect, right? Like,
No, it's
you're
no
not seeing
tail
it.
effect, yeah, which is nice.
How did your presentation go?
For this class? Oh, it was short.
I know, you all told me. 45 minutes, right? What about Melanie? Like you two were on the same day or?
No, other separate days. Melanie's presentation was also, I think, short in the shorter side. I'd say. Our chapters were not that elaborate like you two.
Right.
We had 10 to 13 pages max.
I wanted to join it. At first I thought like I'll ask for a Zoom link. But then I was in a place like a remote place in India where, I don't think so I'll have that network coverage.
Yes.
So I was like, let it go.
Don't feel bad. I thought it was unreal written on your sweater.
I mean,
I was like, unreal?
Whoops.
Today at the elevator, I forgot which floor I'm supposed to press. I was looking like, which floor do I usually go to?
like melon or this one?
This one.
Fours. This is 7, right?
7, yeah.
This is 7, right?
Yeah, this is 7.
Because I
When
stopped
do you
taking
want
the
to give it
elevator.
first or second?
Oh,
It would make
I
more
have
sense
a
about you
sickness.
first,
Yeah.
because part 1, so.
Yeah. I just realized the part 2 was very crap, right? It was nothing in there. Like
Huh?
the part 2 was really mess.
Yes.
Part 1 was kind of like that too. Like they spent 5 pages talking about Drosophila and nematodes. Like I was like, I'm not making slides on this. it was all just to talk about the RAS pathway, just one component of the RAS pathway. And I'm just like, I'm just not going to do that.
So much diverse topics in the part 2, like. But I had a great presentation. Did you guys get the presentation?
Mhm.
yes.
I hope Dr. Kevin comes in.
So today is, if you're done with both the presentations, it's the last day.
What do you mean if we will be done by the both presentations?
You,
There's
never,
no
you
if.
never know.
Today is
I
the
don't think I'll
last.
be that long.
She said that this class will be extended.
But what if something comes up and she says, it's just 30-minute class today?
So I will ask her that if she can grade me without the presentation. then it's fine. I am fine with any grade. Just pass me.
What happened? It was India.
Okay.
Mhm.
You didn't come to school yesterday?
I did.
Where were you?
I was in the admin room,
Oh,
like the room next to Dr. Gleek's office. Like, that area.
the outcomes people, farm administration.
Yeah.
Group.
Because I have to do this.
Right.
That's all.
started. I know. You are that one last night person.
I finished in the morning.
So I told Ashwini that this guy, I think this guy might have slept at 9 and then got up at 3. And then, yeah.
And every time I find articles, I normally find nature articles, big nature articles. Now, what are you looking at? Like smiling.
Of course, you can be a chef.
So why did you withdraw your poster?
I had to travel somewhere. I couldn't be present there. So I said, that's not.
That was a waste of my time actually. And I presented it. Nobody was keen on listening to people from pharmacology.
Really? Everyone was interested in pharmaceuticals.
Yeah.
It's a
My-
pharmaceuticals conference, like.
You won't believe the judge asked me how is the measles virus going to release. Like what? This is not Sotix. I'm not telling
No,
the release profile.
okay.
I'm injecting the measles.
Okay.
Why will
No.
it
Mm.
release?
They are going into that mechanism
It's
of
really
physics.
very relatable. Last year, Rachel told me that after giving her whole presentation, the judge asked, "What is myelin?"
Sorry. Not that one for the students. You can, I didn't have napkins, so you can tear off a piece of these and take a cookie if you like.
Oh, thank you.
Thank
Yeah, sure. Last day of class. And actually, before we get started, and this is not why I brought the cookies, but I do have a favor, or a favor to ask if you are available. My daughter is doing a science fair project where she is trying to determine the effect of color on memory. So she's gonna do like, I don't know exactly how it's gonna go yet, but she's doing like 10 sets of words in different colors, and she needs subjects. So I said I'd ask graduate students. She's gonna come next week, after school, like, I don't know, probably Tuesday and Wednesday. So I don't know if you're around from like 3:30 to 6:30 or so. You'd be willing to help her out.
Sure.
I know Matt, who might not be around, but yeah.
Yeah.
Okay, spread the word amongst your friends because I'm going to
I grabbed 2.
Fine.
sorry, because I'm going to try to email a few people if you know anyone, like if anyone of your friends, other graduate students say they'll do it. I already got OSEP obviously, but if you 5 will do it,
Yeah.
then we have 6. She needs to get 12 to 15, so I'm trying to.
Rachel will do it, like I will ask her as well.
Yeah.
Who?
Rachel.
Oh yeah, ask Rachel. Yeah, Vivek. Maybe you can ask Jeannie.
Yeah.
I will ask her and see.
Yeah, ask around. Okay, so it's gonna be like next Tuesday and Wednesday from 3:30 to 6:30 or so, or something like that. Okay.
Yeah, it sounds interesting.
Okay. Yeah, I don't know exactly how she's gonna do it yet. She won't listen to me. No, she's like, I'm gonna do it my way. She wants to put the words on like index cards in different color, and then show you the index cards, and then I don't know how long she's gonna wait, and then see how many you remember, and then do another color. And I'm like, I think she should put it all like all the words on one sheet, but I'm trying to work her way through it. So I don't know exactly how it's gonna be, but it's nothing invasive. It's literally just trying to remember.
It's
words.
about recognizing the colors.
No, so
Memory.
So it'd be like 10 words written in different colors, like some in blue, like 10 words in blue, and then how many do you remember? 10 words in red, how many do you remember? 10 words in green, how many do you remember? That kind of thing. So,
Interesting.
Because I cannot recognize colors.
oh, that's right.
But I can see colors, like I cannot recognize them.
Oh, you just don't, you know they're different.
Yes, different.
Most of them.
Okay.
But I cannot recognize if someone put a line with a purple marker, like purple pointed, what you call like a marker, thin marker.
Yeah.
I cannot recognize the color that this is purple, but I can see that there is something about it.
Well, it'll be interesting to anyway to have you do it. We'll just write down that you're somewhat colorblind or whatever, and we'll see what the results are. No, that'd be cool. That'd be like you'll be a standalone point, but that would be interesting to see.
Mm-hmm.
Yeah. So yes, please spread the word. Thank you. As long as we have two. Well, my daughter and our Italian exchange student are in the age range. She wants people from like 16 to, I don't know, like 30-ish. How old are you?
32.
I mean, how old?
I'm 32.
Okay, so that's good.
34.
Yeah, that's good. Like that age range, like 15, 20 year age range. Yeah. Awesome. How you doing, Matt? Are you feeling better?
Yeah, a lot better. Yeah.
Good.
That amoxicillin really helped me.
Good.
Matt, download my presentation. Download my presentation.
I actually just replied to like your email
Yeah,
from
you
my to make it easier.
Okay.
Hi everyone. So today I'm going to be presenting the first half of chapter 12, which goes into tyrosine kinase receptors. So the first part I actually want to dig into was actually the very beginning of the chapter, and it went over the discovery of phosphotyrosine. So basically, it was discovered by this guy Tony Hunter, who was interested in identifying transforming antigens of the polyomavirus. It was apparent at the time that infection with the polyomavirus induced an extensive phosphorylation of cellular protein, including the main component of the polyomavirus itself, the middle T antigen. And so what he did was he labeled it with 32 phosphorus, labeled the proteins with 32 phosphorase, and then they were separated by electrophoresis. And then what they found was all of the label was confined to a specific spot, which correlated with phosphotyrosine. What made this story really interesting, why I wanted to include it, was pretty much accidental how they discovered it. Because at the time, it was apparently common practice to reuse buffer for electrophoresis. And so whenever you reuse the buffer enough, it became more acidic. And so if they would have used a fresh batch of buffer, the pH would have been too high, and it actually wouldn't have separated from the phospho-3-amine. And so it was complete accidental moment of discovery. So I wanted to share that just because I found that pretty interesting.
Right, you never know what you're gonna find.
Mm-hmm.
Even like supposedly negative results could lead to something. Cool.
So it was just like a short little story.
That's great, yeah.
I wanted to. And then I also just wanted to briefly lay out, because I also at the beginning of the chapter laid out all of the processes that are mediated through tyrosine phosphorylation. I just listed the ones that aren't actions of growth factors, because whenever I think of tyrosine receptor kinases, I think of like growth factors. But there's quite a bit that's involved with like the immune system, like the activation of B cells, activation of T cells, proliferation of lymphocytes through IL-2, And then in the book, they just said many developmental processes. They didn't say what exactly. They just said many of them. Just closer. And so the first part of the chapter really focused on epidermal growth factor receptors, and then just the ERB-B family of proteins. Now, I've just listed here the ones that they somewhat talked about. So ERB-B1 is just the endothelial growth factor receptor. And then ERB-B2, which is known in humans as HER2, ERB-B3 known as in humans as HER3, and so on. What was really interesting is that HER2 and HER3 don't really act as the receptors. So HER2 lacks the capacity to interact with the ligand because its extracellular domain is fixed in the unfolded conformation, and HER3 lacks kinase activity. They are still important to signaling pathways because these proteins can form heterodimers, and so like the EGF receptor can interact with HER2 and so on. And that's really important because it can modulate the intensity or duration of the signaling molecule. And so the structure of tyrosine kinase receptor, there are two main pieces of the structure that are common across all of the receptors, and that is a intracellular kinase domain, as you can see here in this red, and then a transmembrane spanning domain, which is right down along the middle of the membrane. The extracellular domain varies greatly across all of them. I'm not going to go into all of them because it's a lot, but I included this figure just to show like there's a lot of differences between all of them.
Where's EGF receptor on that chart?
Right here, EGF.
Okay. And where's is HER2 on there?
No.
Just EGF. Okay. I'm imagining they're probably closely related to EGF.
They're probably closely related to each other.
Okay. What else is up there? I just can't read all of them.
Yeah, so there's like EGFR up here. I know VEGFR, but the
VEGF,
yeah.
What's the one with the? It's like 2 or 3 down from EGFR.
FGFR.
That one, yeah. What's that one?
Wait, this one?
No, the
Or-
one above it.
That's INSR.
INSR. I never heard of that one.
So.
Insulin receptor? Or maybe that's insulin
It might be
receptor.
insulin, yeah.
INSR. Okay, I don't know. It's just because and then what's that one with that really long?
PTGF.
ROS.
Ah.
Yeah, I haven't heard of 90% of those.
ROS as in. What's that? Yeah, I mean, I always think of ROS as reactive
Yeah,
oxygen
species,
species.
right,
Yeah,
right.
I wonder what ROS is. Yeah. Okay, thanks. Those are cool. That's interesting.
And then the general feature of tyrosine kinase receptors is that whenever the ligand binds, it causes dimerization. However, there are some like kind of small caveats or exceptions. So some functions of the EGFR can be elicited by cross-linking with antibodies. Platelet-derived growth factor is a dimeric ligand, like the actual molecule is a dimeric ligand, and that actually causes the cross-linking between the receptors. So whereas like we'll get into like the EGFR, the conformation changes and then the receptors itself kind of interacts with each other, the platelet-derived growth factor, the ligand is the bridge, which I found actually really interesting. And then the insulin receptor in the book, it said that it's already dimerized by default, but it still requires activation by a ligand. And to kind of almost complicate matters more, not only do the receptors require dimerization, but conformational changes must occur to increase the affinity of the for ATP within the catalytic domain. So there's a lot going on.
Yeah.
So this is a figure of the EGFR after binding, inducing dimerization. So what happens is that the epithelial growth factor binds to the receptor, and it causes domain 2 of the receptor to kind of unfold on itself, and then it interacts with its corresponding counterpart on its sister receptor, and that's how it causes dimerization. Whenever that happens, the catalytic domain also interacts in induces conformational change, and that's how you get the active receptor. And so there are actually two major domains that predominate in interaction with phosphotyrosine. This is more of like the transcription factors or adapters, regulators that interact with the tyrosine kinase receptor. Two major domains are SH2 and PTB. I included this figure because you can see like SH2 is pretty much in all of them. If not SH2, PTB is involved in some of them as well. So you can see there's a lot of similarities in those domains across all of them, even though there's wide differences with the rest of the molecule. And so this is a figure that shows EGFR in its active dimerized state. And then it's just examples of these binding domains interacting with the phosphotyrosine. So as you can see here, GRB2 and SOS, which we'll get into whenever I talk about RAS. The SH2 binding domain of GRB interacts with the tyrosine phosphase. And then same thing over here with HHC, the PTB domain interacts. So it's just showing what domains of these proteins are interacting with the receptor. And then the book went into some of the signal transduction pathways. First one that they talked about was the PLC gamma PKC pathway. So one of the activities set in motion by activation of the EGFR is generation of DAG and IP3 by PLC gamma. So DAG remains in the membrane and acts as a stimulus for PKC. And so whenever PKC becomes active, one of its first substrates is actually the EGFR itself. It becomes phosphorylated on acerine close to the transmembrane domain of the EGFR, and that actually causes the receptor's inactivation. And then the book said that there's many other proteins or substrates of PKC, but at the time that at least the book was written, there was a lack of understanding of how those kinases determine changes in gene transcription.
So, interesting. So this activation of phospholipase C is independent of G protein coupled receptor activation, right? Yes.
It would appear that, but yeah.
Huh. Okay, cool.
So that was basically the signal transduction pathway was basically just to inactivate the receptor.
Yeah. So it's like a feedback on its own.
Right. And then this was pretty funny. The book spent like 5 pages talking about Drosophila and nematodes. Basically just to get to 2 pages where they talk about the RAS pathway, in which it was perfectly summarized in just this one figure.
Yeah, the book, I've tried to search for a better book over the last few years, and I really can't find one. So this book's pretty good, but it does go off on these tangents sometimes, and I'm like...
Right, there was a lot of words just to describe just this one specific aspect.
Yes.
And it was really the only one that they talked about, which we've actually kind of already gone over in the course, but I'll just kind of quickly review.
Yeah.
So ligand binds EGFR receptors dimerize, and then GRB2 associated with SOS, the SH2 domain binds with the phosphotyrosine, and that allows RAS to be activated by nucleotide exchange on RAS. RAS then becomes activated, and then let's see, where am I? Which allows phosphorylation of MEK. MEK then in turn phosphorylates ERK2. ERK2 dimerizes and then is associated with proteins that take it into the nuclear envelope, which then can go into translation. I think that's what your pathway chapter's on. Yeah. So then that's really the extent of the RAS pathway that they went into in part 1. Unless you wanted me to talk about 5 pages of nematodes.
That's okay.
And then very, very briefly, they did talk about the RAF oncogene. So let me just go back a slide. So CRAF here is the was the RAF protein that they talked about in this pathway, but there are other isoforms of RAF.
Can you go back a second?
Yeah.
What does RAF do in there?
RAF is being phosphorylated by RAS,
Okay.
which then allows MEC to be phosphorylated, then ERK.
Okay. RAF. Why is there no arrow from RAF to MEC?
It's... Yeah, I don't know.
That's weird. Yeah, because that's what I thought. But I was looking at this picture. So what's that?
I guess so. Actually, so RAF comes up to RAS, and then MEC.
Oh, I see it going.
Yeah.
Okay. I was like, well, I knew it was RAS, RAF, MEC, but I couldn't. The RAF in this diagram looked off to the side, but now I see it. Okay, I got you.
They play with like off color, like off red, and
Oh,
like
I know,
red a
yeah.
lot, and
I see
I
it
get
now.
lost
Yeah,
a little
I
bit
see it.
sometimes. But there are different isoforms of RAF: A-RAF, B-RAF, and C-RAF. Mutations in B-RAF apparently have been detected in cancers like melanoma, thyroid cancer, colorectal cancer, ovarian cancers, and they're most commonly like gain of function mutations. What was interesting was the book said that A-RAF and C-RAF are not associated with cancer, and it may be due to just the structure of B-RAF, specifically like the N-terminal region carries more of like an acidic sequence of amino acids, and so phosphorylation may not be needed to confer a negative charge. That was the reason that they gave, but they didn't really go into it too much. It was a very brief paragraph. I just wanted to include it.
Yeah, no,
just
it's
for-
interesting.
So that was everything in my part of the chapter. For my paper, I actually I kind of had the idea of looking for a paper that involves the KRAS mutation for cancer, just because I find that mutation pretty interesting. And while I was looking for something that was a lot more like mechanistic, I came across a actual like clinical trial for a drug that targeted KRAS mutations in solid tumors, and so I was kind of like captivated by it, and I couldn't stop reading, so I decided just to present on it.
Sure.
So, study looked at adacrazib, which is a small molecule that targets KRAS. Here, so KRAS is the most frequent mutated form, RAS isoform in cancer. It has a glycine 12 in its sequence that's substituted with cysteine, which basically prevents GTP hydrolysis, which keeps RAS in its active form despite the receptor.
Isn't that amazing? One residue. One residue. It's amazing that we can even walk and talk, and I mean, seriously, and that there's not more mutated humans walking. I don't know, you know what I mean?
Right.
Well, you know what I mean. I don't know how else to put it, but
Well, I mean, that's why I was looking for papers on KRAS, because whenever I learned about it in my oncology course, I was like, how do you treat a cancer like that?
Yeah, with one amino acid. Yes, yeah.
And then sure
what
enough,
does this do?
the paper didn't really go into the mechanism too much of the molecule, which was kind of a shame.
Okay.
But they did say it was a covalent inhibitor of KRAS, so it irreversibly and selectively binds KRAS. And binds it into inactive GDP state. Study design inclusion criteria was very broad. More than 18 years old, histologically confirmed diagnosis of either like a stage 3B or stage 4, a solid tumor malignancy that had to have a KRAS mutation, obviously. The exclusion criteria was also very, very small. It was just the presence of active brain metastasis or a leptomeningeal carcinomatosis. I think I said that right. And then kind of like the assessments and endpoints that they were looking for, they were looking at adverse events because this was a phase 1 clinical trial, so they were still worried about any sort of toxic side effects. And then their main endpoint was baseline disease assessments. There was an initial assessment performed within 28 days of starting the drug using either CT scan or MRI, and then assessments were performed every 6 weeks after the first dose.
Can you tell me what's leptomeningeal carcinomatosis?
I don't know, and I didn't look it up.
All right.
If I had to guess,
Meningeal. So usually so maybe something with the meninges.
That would be my guess. That
Yeah.
was going to be my guess.
Yeah.
Um,
Yeah.
especially because it was like either active brain mets. I feel like it's anything
yeah,
active
it
in
makes
CNS.
sense. Yeah.
And then they also had a dose escalation segment at the very beginning of the trial to look at the pharmacokinetics of the drug. And so it began with an accelerated titration design, but after a predefined degree of toxicity was observed, they switched it to a modified toxicity probability interval design. I didn't look too much into what those mean.
It probably just means something about they noticed some toxicity, obviously, and then they instead of just escalating the dose without any regard of the toxicity, they probably now somehow added the probability of that
Right.
patient getting toxicity into the study.
That makes sense with the results section.
Right. Okay.
That we went into a little bit. And then they defined the maximum tolerated dose
30%
was associated with
okay.
30% of dose-limited toxicity in 30% of patients.
Really
And then the starting dose that they chose was based on 28-day dog toxicology studies.
Mhm.
And so this is kind of like a graphical representation. So first part, this is right here. Phase 1 is like the dose escalation phase, was with the first couple patients that was enrolled. And then they did expand the trial as it went on. You can see it was started at like 150 milligrams once a day, then 300, 600. It even went up to 1200 once a day, and then also 600 twice a day. And so getting into some of the patient demographics, overall, just kind of like my observations of it, it was a pretty kind of standard patient demographic in terms of like a clinical trial. Age range kind of makes sense for me looking at cancer. Pretty good split, male, female. Ethnicity was not very diverse. The ECOG PS is just like a performance score for how severe the cancer is affecting someone's like quality of life. And so a score of 0 or 1 basically means that they're still functioning most day-to-day activities on their own. Grade 0 is as if like nothing wrong almost. Grade 1 you start to see some limitations, but everyone was within those first 2 grades. Above that you start getting like more of like you're starting to have trouble to do daily tasks. Grade 5 is I think just death on the scale. Smoking history, considering that most of the tumor types is non-small cell lung cancers, this kind of makes sense to me. I was interested to see that they had other types of cancer. They weren't just focusing on non-small cell lung cancer. There were other types of cancer involved, but considering most of the patients were stage 4, I can kind of easily understand why they would be eligible for the trial.
Yeah.
And then this was also pretty interesting, the number of prior therapies before. A lot of them had failed multiple therapies. One patient even failed 8 therapies prior to this trial, which is a lot.
Yeah. Oh boy.
So the summary of the pharmacokinetics of adagrasib, I wasn't going to go too much into it, but the key takeaway that I found was that the researchers found that 600 milligrams twice a day was the dose that had to like maximize the concentration but minimized like without exceeding the dose limiting toxicity limit that they put in place. And so 600 milligrams twice a day was kind of the standard thereafter for everyone entering the trial. And then this table just summarizes the treatment-related adverse effects of adagrasib. Overall, even though there are some very high percentages here, I would say this drug is fairly well tolerated. The most common adverse reactions were nausea, diarrhea, vomiting, fatigue, which obviously aren't great, but in the context of therapies for cancer, basically all of them do that. And so that side effects you can kind of you can deal with those side effects. There's drugs that help with those.
What was the most severe? I can't.
Most
Or most concerning, I guess.
most concerning, looking at this, so they also, this was any degree, these first two were any degree of the side effects, and then these two columns are that were considered grade 3, so serious enough that it required some sort of intervention. Fatigue seemed to be the most common for the grade 3.
That's normal.
And you can kind of see it whenever we get into the results, where some patients had to have interruption of therapy. You can kind of probably see that in the results, even though they didn't specifically tie those patients. Like, they didn't say those patients, but that will be my guess.
Right.
It'll make sense whenever.
Can you read some of those side effects? Decreased weight.
So, other than nausea, diarrhea, vomiting, fatigue, you have a lot of your laboratory panels, so like your serum creatinine, AST, ALTs.
What's on that? Does this say decreased weight on that top?
Yes,
Right.
decreased weight.
Okay. What's under that? Decreased weight.
Dizziness, upper abdominal pain, amylase, dry mouth, dyspnea. One that I found actually really interesting and potentially concerning was QT prolongation,
Oh yeah.
which is the QT interval of the cardiac EKG. If that becomes severe enough, it can lead to torsades, which is a very fatal heart arrhythmia. The reason why I'm like a little concerned about that is because a lot of patients on chemotherapy are also taking like Zofran to help with the nausea and vomiting, which also the side effect of that drug is QT.
prolongation.
So if you're using combination of drugs that cause QT prolongation, you greatly increase the risk of developing torsades.
Yeah.
But other than that, I would say it's a very well tolerated.
Do you see why I talked about the autonomic nervous system so much? Because all these drugs affect autonomic nervous system.
Yeah.
I mean, all those are in line with that, right?
Yeah, QT prolongation is
But
like
it's
fairly
you was-
common with
yeah.
a lot of drugs.
Yeah.
Even in like working at like a community pharmacy,
Yeah.
counsel a lot on QT
Yeah.
prolongation. And then D-Stable summarize the tumor changes from baseline. That's what they call it, percent change from baseline. I'm assuming they just meant by size because they were looking at CT and MRI scans. Overall, there was an average reduction in tumor size. Most of the patients, as you can see here in this orange bar, is they were taking 600 milligrams twice daily.
Is that each individual patient on that graph?
This is each individual patient. Yeah.
So what does, if it's high?
If it's so, this is baseline is 0.
Okay.
Below the graph means there was a positive change. So size got
Got
smaller.
bigger. Oh, smaller.
And
So you
then
mean positive shows of the
positive
study.
changes was a larger.
Larger tumor. So up is larger tumor.
Yes.
Look at that one patient. You gotta. Sometimes I look at these and I see, if you see an outlier like that, that first patient on the left, you have to wonder what else is going on there.
Right. So that one was interesting because they were the only patient on 1,200 milligrams once daily, and it was also the only patient that had the duodenal So,
A lot of things.
there's
Different
a lot
about that,
different about
yeah.
that one patient
Yeah.
compared to everyone else.
And the ones that had the decrease there, what dose were they on? Most of them? 'Cause
Most of them were on 600 milligrams twice daily, and then most of those, if you look at the stars, most of them had the non-small cell lung cancer.
Yeah. It'd be interesting to have another patient with that same, what was it, adenocarcinoma?
Yeah.
And give them twice 600 milligrams a day to see if it changed anything. Because, you think, oh, well, twice 600 is the same as 1,200 once, but it's not, because the pharmacokinetics and everything will be different.
Right, and then this was the graph in terms of like time changes over time because they test every 6 weeks.
Oh, yeah.
Another interesting was this adenocarcinoma was very very short.
Oh,
It was only one data point. And so,
How
and
many
they already
patients-
had progressive disease.
Did they say
So
that any of the patients died? Did they say in the paper?
there was one patient that died. I think it was that one. So like I mean, I'm like prone to kind of ignore the data point, but
No, it's still interesting. I just
it's interesting. It's just.
It's a sad story.
And then, as you can see here, there's some patients that like this one patient had a sharp decrease and was very stable at the decreases, and then had a sharp increase
Oh, yeah.
around 16 to like 17 months after. This is one of those times where I'm thinking that one of the side effects was one of the severe side effects and
Yeah.
required changes in therapy, which caused that. That would be my guess because there were a few lines like that.
Yeah, did they do anything with quality of life in these patients, or no? Just
No.
Yeah.
It was just
It'd be interesting also to see their quality of life because it looks pretty effective in the majority of the patients. But I wonder if they're, because a lot of people say no, I don't want chemotherapy because the chemotherapy or whatever, or the drug, whatever, is worse than the disease.
Yeah.
Yeah, I just wondered. Yeah, okay, cool.
But that was basically the whole trial. I learned that actually that drug was approved in December last year
Oh wow.
for KRAS mutated solid tumors. And that was the actual trial that the FDA used to approve it.
Oh, wow. That's interesting.
So, that was it.
Awesome.
So was it solid tumor or just an SCLC? Because like I've seen such studies where they conduct bucket study where they include all the solid tumor patients, but the last slide that you showed, it is like more beneficial in NSCLC, less in the other solid tumor forms.
Right.
So is it like a broader approval or just
I only very briefly read the FDA kind of like press release. I think it is a little bit more broadly. I don't think it's just small.
Because what I've seen usually with these kind of studies, they include all different types of solid tumors,
Right.
but then they expand their study ultimately into one solid tumor type.
Right. Right. I have to double check that because I just saw that was for a
Do you know where this study was done? Where these authors are from?
The authors were from California.
Okay. It's really fast that it got approved because this was phase 1B,
Yeah.
and this is a 2022 paper, and it got approved when?
December 2022.
Wow. Well, I guess with patients that have very limited options and
Right.
you see some good results, you run with it, I guess. Yeah.
It needs something to hang it.
Yeah.
Especially for a solid tumor with KRAS mutation.
Sure,
because
sure. Yeah,
there's
there's
no
nothing
other
else.
drug class that will work.
Yeah, there's a
for that.
big there's what's it called in the cancer community, like a whole section. RAS is considered undruggable. For a long time, it was considered undruggable. So it was like the people studying cancer pretty much formed this own group of like let's figure out how to target RAS. So it's been on the radar of people studying
Right.
cancer for a while, and I think they just grab anything they can and help because there is nothing else. Yeah, there's nothing else you can do.
Yeah, it says here that FDA granted accelerated approval for this drug.
Expedite it.
Yeah, and it is for like non-small cell lung cancer.
Yeah, it's for
Yeah. Stage 4. Yeah. Well, let's hope that it gave them good quality of life. It's great if these drugs can extend your life, but then you got to think about what's the quality of that
Right.
life. Yeah. Oh, thank you, Matt. That was really good. Good job.
I don't know the years. It should be like a good month. It will be the second one. Yeah.
Is it a full screen?
Yeah, whatever you want. That's not the full screen, but you could do the full. There. Oh, what the heck? That's good enough. Yeah, you can see it, right? Yeah, that's good.
So, hello and welcome to my presentation. And I will be presenting the next half of the chapter 12. And thank you, Matt, for giving me that brief background, and then I can just move on with it. So, the chapter topic in the chapter subject is signaling pathways operated by receptor protein tyrosine kinases, and we will be talking about various topics in this presentation, and it will be more about the next phase of where we move on from like ARC and the substrates of ARC. And the topics of this presentation would be like starting with ARC selectivity. Then I'll be talking about substrates of MAP kinase activated protein kinases. There are many, but I will just be talking about 3 of them. And then how ARC is activating the transcription. And then we will also be looking at the role of ARC in regulation of cell cycle. And then there will be brief discussion about MAP kinase scaffold protein in yeast. And then there is a most important and interesting topic, why these signaling pathways are so complex. So we will be I will be discussing few of the reasons for why they are so complex, and then how we can be how ARC is terminated, what are the pathways, physiological pathways in the body, how it inhibits ARC, and then I will be briefly discussing the family of MAP kinase related proteins, and then I will be presenting a portion of a big paper which is related to combination of ARC and autophagy inhibition in pancreatic cancer. Coincidentally, like it is also related to KRAS, mutation in KRAS. So it's good that Matt has given an introduction to that. So it would be interesting to present that. Starting with ARC selectivity. So every substrate of ARC follows this specific sequence of amino acid, which is serine, urine, and proline. But the problem is all of the proteins which have this specific sequence are not the substrate of the ARC. So how ARC basically specifies on how ARC binds to the proteins or substrates. So ARC bind to its substrates by either binding with a delta domain in the substrate or binding a DEF motif in the substrate. DEF motif is mostly related to only ARC family, not for P38 and junk pathways. So what is a D domain? A D domain is a positively charged residue which is surrounded by a hydrophobic zone. Other than that, what is DEF motif? DEF motif is more related to interaction of ARC with the transcription factors. So there's a picture showing ARC structures, and as you can see in the ARC there are 2 areas. Like one is the common docking site. This is the area where a D domain substrate binds, and most of the substrate binds to ARC in this common docking site. This is negatively charged, and the D domain, as I told you that it is a positively charged, so it binds to this common domain, common docking site. The DEF domain is basically on the other side, and it is related to the transcription factors, and mostly ARC family substrates bind to this area. So protein with a docking motif D domain are normally MEK, RSK, MNK, and ELK1, while DEF domain substrates are KSR scaffolds. We will be talking about the scaffolds in my later part of the presentations, CFOS and ELK1. So ARC1 and 2 and MAP kinase activated protein kinase have a relation, that ARC1 and 2 activates MAP kinase activated protein kinases. There are 5 major substrates of MAP kinase activated protein kinase, which are RSK14, MSK1 and 2, MNK1 and 2, MK2 and 3, and MK5. I will be here discussing the starting 3 of them, that what are they and what are their role in the body. So starting with RSK1 and 2. RSK is normally located in the cytosol, but when it is phosphorylated, it basically shifts partially into the nucleus as well. So in the cytosol, its substrates are factors of the protein translation, apoptosis pathway components, estrogen receptors, guanine nucleotide exchange factors. So its role is, you can understand that its role is mostly related to proliferation, apoptosis, and estrogen receptor activation, and then guanine nucleotide exchange factor SOS. In the nucleus, it basically enforces ERK signaling, like more cell proliferation, by phosphorylating substrates such as ATF4, CFOS, and serum response factor. The other substrate is MSK. Unlike RSK, MSK is only present in the nucleus, and its substrates are cracked histone S3, ATF1, and it helps in modifying DNA and make DNA accessible, basically for more enforcement of the ERK signaling against cell proliferation. The third substrate is basically MNK substrate, which is also called MAP kinase integrating kinases. It's interesting because it is also basically targeted by the virus as well. So starting with MNK, MNK substrate also stimulates protein synthesis, but it phosphorylates initiation factor 4E, which is this, and then this factor binds to a cap of mRNA, and then this activation and phosphorylation of cap of mRNA causes binding of 40 ribosomal subunit, and then starting translation of the protein synthesis, which starts from AUG, adenine, uracil, and guanine sequencing. So this is how MNK basically plays a role in phosphorylating the initiation factor 4E. But there is an clinical application like viruses. As we know that viruses have a habit of capturing the cells and start protein synthesis. So there is an adenovirus which basically hijacks this MNK. It displaces this MNK and hijacks this area over here. So basically they can stop the cellular protein synthesis, and they can start their own protein synthesis, capturing the whole cell from here on.
And this is happening in the cytosol, correct?
This is happening in the cytosol.
Yeah, because of AMR, okay.
Then we'll move on to activation of transcription by ERK2. ERK2 also causes activation of the transcription. How it does is, the upper part of this picture, this story has been told by Matt in the RAS pathway, and then this is the lower part when ERK moves into the nucleus. So first ERK is phosphorylated, and it is translocated to the nucleus. Now this step is quite debatable and controversial as well. Like, it is not clear that ERK stays in the nucleus, or ERK basically translocates, or ERK basically goes back into the cytosol. This is not clear. But after phosphorylation of the ERK2, it causes phosphorylation of ternary complex factor, which is ALK1. And then ALK1 and serum response factor are basically forming a complex over here, which binds to serum response element over here. Other than that, ERK2 also phosphorylates CFOS, and which binds to CJON, and CFOS, CJON basically forms a complex, and then they bind to AP1. This two complexes binding to SRE and AP1 causes cell proliferation. So this is the pathway what ERK does after coming into the nucleus, or it is in the nucleus by default. Then there is a role of ERK in regulation of cell cycle as well. Normally what happens is the cell cycle starts with the expression of cyclin D, which is the main component this expression is required for the start of the cell cycle. After expression of the cyclin D form complex with CDK4, which is required for G1 phase. And then this complex basically inactivates retinoblastoma proteins. Retinoblastoma is given the name to these proteins because they were discovered as in the retinoblastoma, and they were absent in that. So these proteins are named as retinoblastoma proteins, and this leads to moving of more cell cycle progression of the cell cycle. So what ERG is playing the role is the expression of cyclic D is basically facilitated by ERG1 and 2 by induction of the transcription factors FRA1, FRA2, C-Jun and Jun-B. So these are some more transcription factors which are utilized by ERG to basically initiate or start expressing cyclin D, which is the most important expression for cell cycle regulation. Other than that, ERG1 and 2 also stabilizes this complex of cyclin D and CDK4, which is required for inactivation of the retinoblastoma proteins. So this is also helping the cell cycle. And thirdly, ERG1 and ERG2 also inactivates transcriptional co-repressor TOP1. Like TOP1 basically reduces cyclin D expression. So inhibition of the TOP1 also helps the cell cycle to start moving or progressing. So on one side, it is inhibiting the cell cycle repressor, and on the other side, it's facilitating the expressions which are required for cell cycle progression. Then there is a small topic which is related to MAP kinase scaffold protein. So earlier, for the first time, the MAP kinase scaffold proteins were found in a yeast, and they found that MAP kinase scaffold protein having MAP3K, MAP2K, and MAPK in the form of STE11, STE7, and FUS3. As you can see over here, on the left hand side, it is following the whole cascade. That it starts with STE11, which phosphorylate STE7, then phosphorylation of FUS3 takes place, which then in turn phosphorylates STE5, and then in the end it phosphorylates STE4 and STE18. So in this, it basically produces a complete effect, but if this pathway doesn't take place, whole cascade doesn't take place, and only this FUS3 autophosphorylation, so it produces a weaker effects rather than following whole cascade. If it's only phosphorylating itself, it producing a far weaker effect in comparison to when the whole cascade phosphorylates. So they have found this in MAP kinase scaffold proteins.
You say that again? What did you say? What?
If you FUS3 over here
Yeah.
autophosphorylates,
Okay.
it is not phosphorylated through a cascade. It
Okay.
produces a weaker effect.
Oh, okay.
And if it
So it blocks the
yeah,
response?
What?
Do you mean by vehicle effect? It blocks.
weaker,
Weak.
strong, weak.
Oh, weaker. I'm sorry. Weaker. I'm sorry. I was hearing a completely different word. Sorry. Weaker effect.
Weaker,
God.
maybe my
No. It's not you. It's me. I just couldn't.
It produces a weaker effect
Yeah.
in comparison to when the whole cascade is phosphorylated.
I see. Okay.
So this was found in the MAP kinase scaffold in a yeast for the first time.
Okay. Yeah, that's the STE is the yeast protein.
Yeah. It is equivalent to MAP3K, MAP2K, and
Right.
MAPK. Now, the most interesting topic, which I have always amused at why these signaling pathways are so complex. They are cross-talking. They are following so many proteins. Why they cannot jump to one protein and then end up the whole? Why they are following so much? So there were a few reasons in the book which were mentioned. So first of all was a synchronization of transcriptional and metabolic events. So it is also necessary that the metabolic events and the transcriptional events both should follow each other. Like both should be simultaneously. Like one should lead to another. So it should take time rather than going from single protein. It can start the whole metabolic event. So the transcription factors will be far less in comparison to the metabolic factors. So we need time, we need some sequelae to develop before it propagate the metabolic events. Next, the propagation of signals from one compartment to other is also required because there is a distance to complete. Like when the protein or when the stimuli binds to the receptor, that receptor should be endocytized. Then it should it should cover the whole cytosol, come into the nucleus, and then do multiple things. It cannot be done by a single protein. So multiple proteins are required. Then the contact limit of a single protein is very less. Like a single protein can do minimum work. Like maximum, it can bind to few substrates or few things. It cannot do whole of the stuff. So it requires more of the protein to do various functions. Then amplification of the signal as seen in the scaffold protein. As we have seen in the scaffold protein, there were amplification of phosphorylation was more when the whole cascade phosphorylates. If only one protein is phosphorylating, the effect will not be as much as you see in whole cascade phosphorylating. So amplification is also required. And the main thing, pathway should not rely on a single protein. If it's relying on a single protein, and if it goes away, if it produces mutation, then what? So this is also the reason why they are so complicated. Then the feedback circuits are also required for balancing the outcome. Like you need to the on button and the off button both in the body. So if you have a single protein, you cannot have that. So you can the body can also turn on and turn off. So it requires a lot of proteins and complex.
Yeah, it kind of makes sense if you think about it, because these events are so important and so varied that the kinases regulate. So you wouldn't, if you just went right to the kinase, there'd be no way for the checks and balances and amplification and all that stuff.
Then next we'll move on to termination of arc. Arc is also required to be terminated or attenuated because we have to control the effect of arc as well. So how it can be done? It can be achieved by 3 steps or 3 ways normally, as the book has mentioned. The first is the stripping of phosphotyrosine residues to inactivate arc. Most of the phosphatase causes basically stripping of the phosphotyrosine residue, which leads to inactivation of arc. Those phosphatase are PTP1B, SHP, DEP1. And then the second way can be the suppression of RAS pathway. If we suppress the RAS pathway, ARC cannot function. So suppression of the RAS signaling pathway by phosphorylation of guanine nucleotide exchanger factor. It can also be done by it can basically inhibit the activity of the ARC if we suppress RAS. And then the third is removal of the cell surface receptor by endocytosis. If we can remove the if the body can remove the receptor, or it can basically endocytize the receptor, then the ARC effect can be reduced. It is shown in a picture over here, like how it is done physiologically. As you can see over here, this is EGFR receptor, and it is ubiquinated, and there is a CBL handle which basically binds with the ubiquinated receptor, and on the other side it binds to endophyllin and activates AP2 to endocytize the receptor in this way. So once the receptor is endocytized, the activity of ARC is basically inhibited. But one more thing, until the receptor is completely degraded by the lysosome, it continues to produce the signals. It continues to produce the effect. So degradation of the receptor by the lysosome is also required to completely terminate the effect of ARC.
And is it only, to your knowledge, only the dimers and the activated receptors that are endocytosed?
First, it is required to endocytize the activated receptors because they are the one which are the most concerning.
Right.
They should be stopped first to
stop the action, right? Yeah. Mm-hmm.
Then, slightly briefly about the family of MAP kinases. You know that ARC1 and 2 are the classic MAP kinases. And then there was information of ARC3 and 4. And whenever I read this, I just get the question of Young. in Asif's presentation that what ERK3 and 4 are,
Yeah.
why you are not targeting it. So
There's
why
not much known, yeah.
yeah, why they are not, why it is not targeted is because mainly there is no tyrosine residue in the active segment of the ERK3 and 4, and we don't know much about the downgrade substrates and downstream substrates and other effectors of the ERK3 and 4. So targeting them is not of any use, and they have a very different functions and very little is known about them. So, but ERK5, which is the favorite for Dr. Kavanaugh's lab, and this ERK5 is quite similar to ERK1 and 2, except that ERK5 has a big size of protein, and ERK5 is also called as big map kinase, BMK1, and they have similar substrates to ERK1 and 2, and they can directly bind to DNA. Other than that, they are mostly involved in the development of CVS and neuronal differentiation. So ERK5 has are similar to ERK1 too, but slightly different functions.
And more recently, all implicated in cancer. So survival and proliferation, ERK5. ERK1 too a long time ago, but ERK5. So ERK5 is called big map kinase because it's a much larger protein than ERK1 and ERK2 and has this unique C terminus that can directly bind to the DNA. It acts as a transcription factor in and of itself. It also autophosphorylates itself. So we're learning more and more how different it is from ERK1 too, but they do have a lot of overlapping roles in the cells and such. Yeah.
There are two other pathways as well, JNK and SAPK, JNK pathway and P38 kinase pathway. They both are stimulated with the stress. Like other MAP kinase activated pathways are basically induced by mitogens, but these are activated by stress and cytokines. And in JNK pathway, CJUN is phosphorylated, and P38 kinase, HOM1 expression is induced.
Oh, also, my lab, we might, and along with Dr. Flaherty, might be looking at ERK7/8, which is MAP kinase 15. That not a lot is known about. And ERK6, I think, is actually a P38 isoform. So there's some things that are like, yeah. Some
The
things
book is too old.
I know. That's another reason I was looking for a newer book, but it's just, yeah, the book's too old for that. Yeah.
That's why they have not mentioned these things.
Yeah.
And there's very little knowledge about these as well.
Yeah, I know.
The literature has moved a lot forward.
Yes, yeah.
Then this is a classical pathway which we normally see.
But it has ERK5 in it, so it's not that old. I mean, a lot of textbooks still don't even mention ERK5, which is, I'm surprised.
So this is a book which is way ahead of its time.
Yes, yes, that's right.
Like ERK5 is relatively newer.
Newer, yeah. It's now 25 years or so, but that's new compared to ERK1, 2, P38, and JNK.
Yeah, so all these pathway end in activation of MAP kinase. Other than that, ERK pathway and JNK P38 pathway crosstalk as well. But JNK and P38 pathways are activated by stress cytokines, and these ERK pathways are activated by mitogens.
By the way, it all, like stress and cytokines can also activate the ERKs now, we know that. Mitogens can activate P30, it's all over the place.
So is the pathway like either ARC or JNK, or can both of them like get activated simultaneously? Okay.
They both can get activated simultaneously. Yeah.
So moving on to the article,
Cool. What year is this?
2019.
19.
Yeah. So what we have, I've chosen this article because it was related to ERK as well, and autophagy, which is a topic which I like a lot. So in this article, combination of ERK and autophagy inhibition is checked in the pancreatic cancer. And this pancreatic cancer is basically pancreatic adenocarcinomal tubular cancer. And in this type of cancer, KRAS is mutated, and KRAS is one of the potential targets, as mentioned by the FDA, who like they're targeting the KRAS or suppressing the KRAS would be like beneficial in the outcome of pancreatic cancers. So moving on to the introduction, the pancreatic ductal adenocarcinoma depends on KRAS and autophagic flux for its growth. Like KRAS basically provides the nutrients to the carcinogenic cells for the tumor growth. And the other part, like other thing which provides the nutrients, is autophagic flux. What is autophagic flux? Autophagic flux is basically degradation of the bigger organelles of the cells to produce byproducts, and from those byproducts, the energy is produced to provide the nutrients to the proliferating cells. So once they are the energy source, this autophagic flux is basically the energy source for this type of cancer. So, and what is RAS? KRAS? KRAS is gene of RAS family, and it mediates RAF, MAC, ERK pathway as well, and P13K, AKT, mTOR pathway. Mutation of KRAS is associated with PDAC, non-small cell lung cancer, and colorectal cancers. And as I mentioned, that autophagic flux is a process which produces energy by degrading the organelles in the lysosomes. So what was the aim of the study? The aim of the study was to evaluate the impact of KRAS and ERK inhibition on autophagic flux. Like what happens when you inhibit the KRAS or inhibit the ERK on the autophagic flux, like is it increasing? Because once you inhibit the energy supply from the KRAS, obviously it will have to increase take the energy more from autophagic flux. The next was to determine the effect of blocking both ERK and autophagy on PDAC. If both the supplies are cut for the cancer cells, then the PDAC cells, like pancreatic ductal carcinoma cells, are like decreasing or not. So results were, first they look at KRAS suppression elevates autophagic flux, which makes sense. Where if you stop one of the supplies, the other supply will increase. Like say what they did was they took pancreatic duct cell cancer cell lines, and they infected them with lentiviral vector encoding EGFP, LC3B. LC3B is an autophagic marker, and then they also transfecting with siRNA for KRAS inhibition. KRAS inhibition was done by siRNA. So if you can see in these all cell lines, the autophagic index was basically increased with the inhibition of KRAS through siRNA. And if you see in this Western blotting, the LC3B was basically, B1 was converted to LC3B2, which is autophagosome marker, and it basically indicates that autophagy was basically increased with the inhibition of KRAS. And they also did one more thing. They used baflomycin A1 to inhibit autophagic flux. With the inhibition of the autophagic flux, you can see that the conversion was decreased, and without the inhibition of the autophagic like flux inhibitor, the conversion was increased with the inhibition of KRAS. The next thing was inhibition of ERK1 and 2 increased autophagic flux. Similar result was found because ERK1 and 2 and KRAS works in the same manner. So inhibition of the ERK1 and 2 will also decrease the supply line from the KRAS. So ERK1 and 2 was inhibited by this product, SCH772984. I don't know that much about it, but it was inhibited by this product. And then again, same thing was done. siRNA. It was like autophagic flux index was checked, and it was seen that it was increased in all the cell lines with the inhibition of ERK1 and 2. Similarly, as we saw with the inhibition of the KRAS. Again, the autophagy was also checked, and with the inhibition of ARC, we saw that conversion of LC3B1 was increased to LC3B2, and we found a similar result that inhibition of either ARC or inhibition of the KRAS produces increased autophagic flux. Then the third thing, inhibition of ARC and autophagy together basically reduce PDAC proliferation, which is pancreatic ductal adenocarcinoma. And we saw that in these 3 cell lines, SCH was basically given to inhibit the ARC, and CQ is chloroquine, which is autophagic inhibitor. And as you can see, with both given together, in all 3 cell lines, it is producing a synergistic effect, and it is decreasing the PDAC proliferation together. Then they also checked this synergistic effect with inhibiting the multiple levels of autophagy. Like autophagy is done in a sequence with the different ATG1 to ATG2, 3, 4, 5, 6, 7. So they saw that with inhibiting the autophagy in different levels also causes, with synergistically, it also inhibits, it decreases the PDAC cells through inhibition of the ARC. So if we see that inhibition of ARC was done in all the cell lines with different concentrations, and multiple levels of ATG were basically inhibited. As we see that most levels of the ATG were inhibited with ARC was producing the better result. So if we inhibit the ARC and autophagy both, we'll get the best result.
So they're all different. Are they different cell lines? Is that what that is?
Yeah, these are all different cell lines.
And then, what's the difference between the top and bottom graphs? I'm sorry, I'm not seeing them. Oh, eight.
Different cell lines.
Different cell lines, and then different com.
Different levels of the autophagy. Like these are autophagy 5 to 1, 5 to 2. These are autophagy 7 to 1, 7 to 2, 7 to 3. More of the sequencing of the sequelae of the autophagies are inhibited.
Oh, okay.
So if you inhibit autophagy at multiple levels, you will get more result, like better outcome.
Okay.
Because autophagy basically produces the products which provides the nutrients to the cancer cells.
And what's NS?
NS is basically a control for I just don't remember.
Normal saline.
No, it's not normal saline. It's not normal saline.
What is
I just didn't.
Because NS looks pretty effective at higher, you know what I mean, at higher concentrations anyway.
Yeah.
I mean that first.
I don't remember the full form of the NS. NS was basically.
Is it a positive?
No, it's not a positive. It's not a positive control.
No, because you see what I'm saying.
Yeah, it's in the end. It's basically.
Yeah, and even in that first top left graph, A.
This one, A.
Yeah,
it's finishing over here.
Yeah,
but it's better than the ATG5 at the end.
Yeah.
Yeah.
Yeah. So I just wondered what the N is.
So in conclusion, KRAS suppression elevates autophagic flux. Inhibition of ARC1 and 2 increase autophagic flux, and inhibition of both ARC and autophagy reduces PDAC proliferation synergistically. Thank you.
Just one question. I think I missed it. Autophagic flux versus autophagy or autophagy. What's the difference again? I'm sorry.
Autophagy flux is basically after degradation of the organelles in the lysosome.
All that.
The byproduct is utilized for the energy.
Oh, okay. All right. I see. So you have to inhibit the entire
autophagy. to
Right.
decrease the energy
You
supply.
can't just, yeah, right. I mean, that makes sense, because how would you reduce the flux
Yeah.
without reducing
The autophagy, yeah.
Yeah. I was looking up this, sorry, wait, that's why I missed it. I was looking up this ERK1/2 inhibitor. It is pretty, because a lot of them are not specific for ERK1/2. This one seems to be very specific. And I just found, actually, I went to Barcelona last summer of 2021. 22, I'm sorry. Summer of 2022, I went to Barcelona for an ERK5 meeting, and I just found a paper from people I met there, and they, with this inhibitor, the S whatever,
Something.
SCH whatever, when you inhibit that in some of these cell lines, what happens is ERK5 compensatorily upregulates and leads to resistance for that
Yeah.
inhibition. So
That makes sense.
yeah, ERK1 and 2 and ERK5 are they're constantly like
Interchangeable.
yeah, and it's funny they call it playing whack-a-mole. Have you ever played that game at a carnival? So it's at a carnival or Kennywood, it's just like it's a game that has a bunch of holes and this mole pops its head up in the object and you whack
Yeah.
it with this big
Yeah.
hammer.
And
then
Yeah,
it
yeah,
pops
yeah.
up another hole, yeah.
Yeah.
Ah.
Yeah, so they're saying it's like inhibiting one, the other one pops up. So then you whack that one and the other one pops up. So it's like, yeah, right.
That's why they are so complex, the
Yeah.
pathways, because
Yeah.
they cannot
Oh.
rely on one single protein.
Yeah, tell me about it. Yeah. So, Patrick reviewed this paper. Interesting. Patrick Flaherty reviewed the paper.
This one, Nature Medicine, which I have.
Oh no, this one's a review in Cell Biology from 2021.
Oh, and the NS in your graph is non-specific control construct.
What's the control?
Yeah.
Oh, so they transfect it with different constructs to inhibit different parts of the autophagy. Is that the idea?
Yeah.
I think so.
Okay, I didn't realize it was so that's interesting. So just the transfection with even a non-specific seemed to like somehow they didn't like it after a while.
Yeah.
Yeah. Interesting. Very cool. My favorite. We could talk all day. I mean, this is right up my alley. So yeah, very interesting. Thank you.
I have a question, ma'am. Like, so if we have to inhibit the proliferation of cancer cells, you have to inhibit ERK1, 2, and 5 together. Then we'll get something.
Yeah, it depends. I mean, so definitely, we found actually, interestingly, in some of our breast cancer cell lines, that ERK1/2 isn't playing as large a role as ERK5, and then in others it's flipped. And we found that with some of our novel inhibitors, the ones that Dr. Flaherty's lab are synthesizing, that you can inhibit ERK5 without inhibiting ERK1/2, and that the cancer cells have a high level of ERK5, so that you can use a kind of a lower level of the drug, and they'll die. But the ERK1/2 is more in the healthy cells, the non-tumor cells, so that targeting ERK5 kind of leaves the non-healthy cells alone. They still die a little bit, but not as much. And then the cancer cells die more. So we're in some cells there is like this dichotomy where you can inhibit one and get a pretty good effect. It all depends on the cancer cell and the genetic mutations and what's going on. Like I said, I think I said this earlier in this class, I think it's going to come down to, with cancer, that we are going to have to genotype every single person's cancer in order to get really effective therapies for every single person, which is expensive, time consuming too, because if you have someone who has a pretty aggressive cancer, you want to start treatment right away, where you could actually be, unfortunately, like accelerating the cancer in some way, right? Because if you target one of these pathways and the other pathway takes over, I mean, at first it's good, but then eventually, right? So, yeah, I really think the only way to treat cancer effectively is going to just be genotyping every single person's cancer, which is not cheap. I'm paying the bills right now, trust me. And a lot of insurances don't pay for that, actually, for the genotyping. Yeah, because I got genotyped just because I wanted to make sure for my daughters, right? Turns out I don't have an elevation in any of the markers for breast cancer, so mine's like sporadic, not necessarily genetic, which is good. I mean, my daughters still have a higher risk, but it's not, at least I know it's not a genetic thing. And yeah, and that bill was crazy, and insurance doesn't pay for it. Isn't that so stupid? Yeah.
I can talk for hours about insurance.
Oh, right. Right. And it's funny, my husband right now is working for a company. It's kind of like pharmaceutical marketing, sort of, but what they do is they work with the providers. So they work with the insurance companies. So they have, and the pharmaceutical industry. So the industry comes to them and says, we need to convince this provider that this is an effective therapy and it should be on their list of approved medications. And he said it's crazy. Like he's like, you have to. It almost seems like it's more than getting FDA approval. Like to get the insurance companies to agree to include these things. It seems like it's really frustrating. It really is. I don't know. I mean, I do think that Obama was onto something with universal health care. I don't think it was perfect, obviously, but I do think he was onto something with that. I mean, I do think that it has to be controlled at that level. That's where all the money, I mean, them and pharmaceutical industries, those are the two, obviously, the two powerhouses. So, it's got to be controlled at some level. And let's be honest, a pharmaceutical company says this drug is, I don't know, something ridiculous. Like some drugs are like $30,000 a year or whatever, or even like $1,000 a month even. Come on. That drug didn't cost that much to make, so the insurance company should be able to work with them to kind of, you know what I mean? I don't know. It's just a broken system. Something needs to be done. I don't know what. I'm not smart enough for that. I don't know the ins and outs of all the industries and stuff, but something needs to be done. Yeah. How is it in your home countries? Like your Philippines, right?
We do have government insurance, and then some people prefer to get their own private insurance.
Okay.
But then we also have problems with coverage.
Yeah. Even by the government insurance.
Yeah, even with the government. So most of the people just get medicines out of pocket. So,
Really? Out of pocket. How about in India and
So India, we have both public sector, public sector insurance and the private
it's the
insurance.
same thing?
But then there are also trusts.
What?
There are trusts, like you have trustees. So fortunately, industrialists they have their own trusts that cover for the, I would call them NGOs, but as part of the CSR activities, they have their own trusts that support below poverty line or below poverty line or just poverty line patients. They support them. For example, the Tata Institute, they support patients with
Okay.
cancer.
So when you say government insurance, is that free to everyone?
Yes, mostly.
yeah, but they still have to apply.
It's more subsidized than.
Okay.
It's not totally free, but
Yeah.
subsidized.
And what about Young? So Korea?
So
Are you from Korea?
yes,
Yeah.
so we are also similar, but we co-pay like a 30%. For the cancer patient, they have probably like if the insurance covers then 20%, so they might
Insurance
have.
covers 20%.
The patient co-pay 30%,
Oh, okay,
yeah, 20%. So some cases, if the insurance doesn't cover, then they have to pay like tons of problem money, right?
So it sounds kind of similar in a way. I don't know. I mean, it's not exactly universal health care like Canadian health care. It's a little bit more universal, right?
My family's British, so my family's British.
Okay.
So they have the NHS. which is complete government-owned healthcare. Hospitals are government-owned. The workers are government employees.
So do they pay?
So they pay taxes,
taxes.
but then everything else is basically free of charge.
You pay
Other
taxes?
than prescriptions.
Yeah, we pay taxes.
Other than prescriptions, but like their prescription, like my grandma's copay for like her blood pressure medication's like 30 cents. So,
It's same. Like my best friend lives in London for like the last 8 years.
Yeah.
So when she had her baby, I mean, yeah, they do pay higher amount of taxes like compared to US. Their
Yeah.
tax is like unbelievable right now after
Right.
COVID. But well, when she had 2 babies, she didn't have to pay anything out of the pocket other than some small medicine drugs. Like everything was free.
Wow.
And even they sent her home with like 2 weeks of antibiotics and all that. Like these, you have to continue this.
Yeah, and they have a year off at least from when they have a baby and all that stuff.
Yeah, she could
Don't even
like
get me
she
started
worked
on
from
that part
home
of it.
for a lot of time.
Yeah.
So that was good, but what she said was the downside was right now the tax is unbelievable.
Yeah.
Like they cannot think about one earning, like both of the parents had to earn if they have to pay.
Yes, yeah.
Another thing is the doctor's appointment is very like.
That's what I was gonna ask. Is
Yeah,
it really hard?
It's
I mean, it's not that easy here, to be honest.
right.
Yeah.
The problems with the NHS are a lot more on the provider side. So like doctors are overworked, underpaid.
Yeah.
I could talk for hours about this too. It's mostly from the actions of one political party in the UK
Yeah.
that wants privatized healthcare.
Uh-huh.
But yeah, junior doctors are basically paid minimum wage for cleaning out work. It's crazy.
Wow.
It's something like that in Bangladesh as well. Like we have government hospital where everything is cheaper and most of the government supply drugs are free, but the doctors over there are almost overworked. And sometimes they have to work for 16 hours straight, which is just unbelievable.
Yeah, right, right.
Yeah.
It almost seems like all of this in every country needs to just like be wiped clean and start new. Something. I don't know what it is. I don't know the answer because I don't know. But
And
there
somehow
needs to be
this is supposed to be a care institution, but it has become a business model
Yeah,
for
yeah.
so many organizations that
Yes,
the
yes,
care
yes,
portion of it comes at the very last.
Yeah.
Right.
Well, I mean, I see my husband, and you know, they work, like I said, for pharmaceutical companies, and then to try to get the insurance people to pay. So, as part of the cost of this drug, right now he's working on a muscular dystrophy drug, and it's like astronomically expensive because there's no medications for this, and I think it's an antibody, some kind of immuno medication or something. Ends in mob, right? Yeah, so, but like, just the amount of hours he himself is working on this, and I know what he gets paid, and he gets paid a nice salary, you know, to do this, and they bill it to these pharmaceutical companies. So that's part of the cost of that drug. So when you see just this little snippet, like that's not even the production,
Yeah.
the research, this, and how much they make, that the company he works for, not just him individually, but I mean the company he works for, how much money they bring in. Like no wonder drugs are so expensive. You got to spend all this money just to convince insurance companies. Like I almost think like if the FDA approves it, why wouldn't you provide that medication for your patients? You know what I'm saying?
It's really frustrating at Port Medical Pharmacy because I mean one drug that like irritates me that insurance companies don't cover more of is Eliquis, and basically all of like the Dolex, the newer anticoagulants, they all will cover warfarin at no cost, but they won't cover like Eliquis.
But warfarin, the warfarin's the one where you got to keep going in. and
Exactly.
get those blood
It's like you
tests.
gotta get iron R's checked.
So it's more money.
It has a
to
lot
take
of
care of
drug
that patient.
interactions,
Right, yeah.
like, but whereas you take Eliquis, it's just 5 milligrams twice a day
Right.
for basically
And you
everybody.
don't need all that blood work.
And you don't need all the blood work and everything, and it
It's
works.
safer. Eliquis is safe. Is it the rivaroxaban or apixaban?
Eliquis is a Pixabay.
Apixaban. It's much safer.
Yeah.
Way
It's
safer.
so frustrating. I think if a drug's approved by the FDA, you should be forced as an insurance company to cover some of it at least a portion of it. You know what I mean? Like, That's crazy.
So when did you start having this problem? Was it with Obama? Or did you have it before?
Oh, we had
Oh,
it
it's
before.
always-
I think Obama care made it maybe a little bit better. It was a hopeful thing that he was trying to do. I think he was trying to do the right thing, but I don't think it worked out that way necessarily.
A lot of it was blocked.
Yeah.
A lot of what
yeah.
he wanted to do was blocked.
So unfortunately, but
Of course.
yeah. Yeah, it's because actually even just with me, which is a fairly straightforward, luckily low risk case and everything, I was talking to my doctor and he said something about we were trying to decide which hormone, which hormone therapy I would take. Would it be an aromatase inhibitor or would it be estrogen receptor inhibitor? And we were naming some different things and different side effects and this and that, my age, bone health, blah, 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 blah. And he's like, wait a second, what insurance do you have? And I said, UPMC. He's like, forget it, we can't even talk about that because they don't cover it.
Yeah.
So then we had to go back to the drawing board and I was just like, what? He's like, yeah, they don't cover this and that. Like even UPMC, you think like, it's good insurance, I'm not complaining, but like,
What they say goes.
yeah.
So that's the first question you should ask.
Yeah, is this covered by
It
insurance?
is.
Yeah.
It is legitimately the first question.
Yeah.
I've been thinking of that.
Yeah, and which is sad because you want the best therapy for whatever you're dealing with, right? But you can't always get that, you know?
You have to ask what insurance you have just to know what pharmacy to go to.
Yeah.
Because
Oh, right now.
this year,
and that's-
CVS, you can't go to CVS if you have UPMC.
I know, and it's Rite Aid, and now Rite Aid's
Right.
like basically
And Rite Aid
going
is
out of business.
imploding. Yeah.
Yeah.
So now what am I gonna do? Because it's not Walgreens. I don't think Walgreens.
Giant Eagle.
Giant
Yeah.
Eagle. Okay. Yeah. Okay. So I don't think Walgreens takes UPMC.
I don't know, but Walgreens is another one that's kind of falling apart.
Are they really?
Yeah,
I
there's like
Walgreens.
there's a Walgreens that I drive past on my way to work. Closed every weekend. Like not open on weekends. Very limited hours now, just because they don't have pharmacists staff.
Why? We're producing a lot of pharmacists. Actually, fewer now, but I don't know. Everything's going in the wrong direction. Not to be doom and gloom, but I feel like everything's going in the wrong direction. I mean, we might have a criminal as president soon, so I mean, what is going on? Like,
Should I go home?
What?
Should I go home now?
Yeah, I know. Well, it's funny, like, even with all our problems, apart from Donald Trump, I'm just going to put it right out there, becoming president again, if he does not become president again, I still think this is a better country with a lot more freedoms and things like that than a lot of other countries, okay, yes. But if he becomes president again, all bets are off. I would maybe go home. I'm not kidding. I mean, he's dangerous.
It's crazy that after all that he's still
Yeah.
a nominee candidate.
Yeah. What?
It's just
Why? What? Who are these people? Who
Was
are-
it that bad in that area, in that time period?
Yes. 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 Yeah, he's just